Good morning to all. I am S.Santhini, Department of Biochemistry. Now we are discussed about plant biochemistry in plant tissue culture. What do you mean by plant tissue culture? Plant tissue culture, it is always known as the test tube will be always treated in a test tube, and then always known as the plant tissue culture is always known as the plants to be cultured in a test tube due to the used as a nutrient medium is always known as the in vitro lab method. What do you mean by in vitro and in vivo? In vitro method is always known as the laboratory method. In vivo it's always inside the living organism. Next we are talking about plant tissue culture. The first one is the main principle of plant tissue culture is here. Two things, one in totipotency and plasticity. What do you mean by totipotency? It's always known as the culture of plants in a test tube, then always plant it into a regenerate, then the plant to be growed in a perfect environmental and greenhouse. Next, plasticity. What do you mean by plasticity? The plants always grown in a test tube. Next, they always regenerate and a multiple clones of plant to be produced in a culture medium due to the micropropagation. What do you mean by micropropagation? The micropropagation is always known as the culture of plant is always grown, that it's always normally healthy plant, then always regenerated the plant materials. Then first step in plant tissue culture to be selected a explant material. What do you mean by explant? It means the plant either are the plants organs, tissue, cells, seeds, etc., they are always to be taken, then it's always known as the explant material. Then how do we sterilize the explant material? Sterilization, the various type of sterilization technique always used as the plant tissue culture. In example, tri-heat sterilization treatment, it's always known as the tri-heat treatment. Next, autoclaving, wiping at 70% alcohol. Next, the transplantation. Okay, then the sterilization technique. Next, the particular plant to be explant. Next, selected the explant material to be selected. Next, the sterilization is another one. The plants and the equipments, vessels, instruments, and all of in overall laboratory to be sterilized. Next, in sterilization, next the particular explant material due to the third one step is inoculation. The particular explant, look like a organ, the plant tissue or organ to be taken out, then they are sterilization. Next they go to the another level to incubation. The particular plant material to be incubated at particular temperature or Celsius. Next, the incubated plant material was to be collected into a test tube. Next, to produce a culture medium. The culture medium is also known as the nutrient medium. It's always known as the either or the agar material, the agar medium are always known as. Then adding a plant growth hormones. What do you mean by plant growth hormones? The plant growth hormones is one of the particular healthy plants to be growed in a particular medium to healthy plants, because the various types of plant growth hormones to be used. Axine, gibberellin, abscisic acid, cytokinin, etc. In plant tissue culture, separately axine and gibberellin to be used as a plant tissue culture. Next, the first one is explant is over. Next, the explants to be sterilized by autoclaving. Next, then the sterilized plant material to be incubated, then inoculated into a callus culture. What do you mean by callus culture? The callus culture is divided as a particular plant material, look like a tissue. It is tissue too. Separately cells are divided, then to be grown in a particular cells, because the cells are cell divide, then grow a daughter cells, because the parent cells are always cell divided, then to produce a new cloned plants. The main aim of plant tissue culture is always produce a cloned plants. Next, first explant, sterilization, incubation, inoculation of subculture. Next, the another step is very important to inject the plant growth hormones. Next, inoculation. The inoculation is a various technique to produce a subculture. Next, again the plant material to be regenerated to produce a newly synthesized plant, cloned plant. The cloned plant is large variety of new cloned plants, then always taken into the transplantation. What do you mean by transplantation? The healthy test tube plants to be transplanted into the new atmosphere, greenhouse, house area in other street and native places, etc., etc. Then the main advantage of transplantation technique is used to aim as there are lots and lots of new plants to be varietily to produce the plant tissue culture technique. In the season, the particular season, the plants would not have able, but plant tissue culture technique is always advantage to produce the new plant tissue culture, new plant tissue, new plants. Okay, then the main advantage is plant tissue culture is multiple clones of plants. Thank you very much.